let's get started. So where we left off last time, just to recall, we were finishing up the genetic algorithms. So we've, and I'll kind of review up to where we were, but we're about to introduce the crossover and mutation operators, which will then close out the rest of the algorithm. So just to recall, we have this decision vector that I've been calling, X vector here, and that we're often referring to as a chromosome. And each component so X1, X2, etc., is a gene. So genes are just locations along these chromosomes. So we might have N genes, and that would mean that our decision vector maybe comes from Rn. And so each component we might also call a decision variable. And so then particular strategies that we're testing out so if I have a particular strategy like equal to, I'll go with a subscript of 1, and maybe it is some, sequence of numbers here, just some vector. And so what is the name we use for a particular strategy, a candidate strategy? So if the decision vector is a chromosome, and if positions along the chromosomes are genes, genotype.
Yeah.
Okay, good. So this is the genotype of a particular individual. So we can think of this as an individual in the population, and we can think of this as the individual's genotype. So I'm an individual in the population, I have a genotype, you all have your own genotypes, we have some variance in our genotypes, but just because we have variance in our genotypes doesn't mean we have variance in our fitness, and that's what we'll get to next here. So I'm going to make this a little more formal. This will help set us up for when we jump to multi-objective genetic algorithms. So we're going to take a sort of we'll diverge a little bit in the next couple lectures, and then we'll come back to multi-objective genetic algorithms. And so in prep for that, I'm going to try to move us along from thinking, right now I think everybody's got this like one-to-one correspondence between a fitness or an optimization objective and a fitness function. And in reality, you can have multiple optimization objectives, but you always only have one fitness. And so there's a slightly more complicated mapping there. And so I'm going to introduce a few other terms here. So our optimization objective, and in this case, we're going to say we just have one, but later on we'll have multiple simultaneously. And I'm going to call that a character. And what do I mean by a character? I want to say that, for example, eye color is a character. And so if you have an optimization objective, it might give you a particular eye color. And that particular value of the character is known as a trait. So a particular value is a trait. So right now, what the optimization objective does is it maps genotypes to traits. So we have F genotype ends up mapping to a particular trait. So an individual has a genotype, and that individual has a certain trait value. So my genotype might give me, well, combined with my environment, my genotype might give me brown eyes, your genotype might give you blue eyes. And so this, those particular values are there in these traits. Now what fitness does is it then maps those traits to the number of offspring you have, or something that correlates with the number of offspring you have. So the fitness function which I'm going to use as a capital F, effectively maps traits, plural. So, you know, this is just emphasizing that this will in general be plural, to the quality, to something that relates to the quality of the suite of traits. And this is generally proportional to number of offspring, at least on average. So right now, we're only dealing with a single optimization objective. So it looks like we've added a whole lot of indirection here for no good. So I'm saying that we've got from genotype to trait, from trait to fitness. So there's only one trait here. So for now, we are, you know, for the single objective case, so for single objective, then there's just this composed function which is mapping genotype to fitness. And so you can think of this as quality. So you have some optimization objective, little f, that your boss has given you, or the problem in general has given you, and that optimization objective could be, you know, positive numbers, negative numbers, or whatever. But then you're going to say, I want to minimize that optimization objective. Well, then you encode minimization into your fitness mapping, or you encode maximization into your fitness mapping. Or you might say, I want to get this optimization objective as close to a particular value as possible, where you would encode that into your fitness function. Things get more exciting when you have multiple objectives. When you say, well, for a particular decision vector, you know, if I make these design choices, I get a really fast robot. But that fast robot uses very expensive parts. So I can imagine I have two traits, the speed of the robot and the cost of the robot. And if I care about both of those, then somehow this fitness function will encode how I care about both of those simultaneously. But for now, it's as if I only care about speed. And so there's a number of different parts that go to the speed. Maybe the weight of the robot, you know, that's maybe a decision variable. Maybe the types of motors I use, the size of the motors. So those all might be decision variables that are genes that go into the chromosome, and then the particular choices I make are the genotype, and then how well the robot does versus other robots is the fitness. And so that's the mapping there. Is there a question?
Yeah, I had two questions. Number one was that I don't understand what you say by character over there. And number two is that, so we are going to optimize the small f of x.
That's the
right?
goal, yes.
And there would be multiple such traits, right? So there might be multiple such functions that we need to optimize with that one fitness function.
Right.
Now, if we don't know the relationships between each of those individual traits, then how do we know what to put in the fitness function?
Right, so that we'll get to that when we get to multi-objective optimization, but in short, cutting to it, there's of course, if you know the relative weightings, it's easy. You can scalarize the whole thing and add all these things up together. But in general, in multi-objective optimization, we leave that scalarization to the stakeholder to do later, and what we actually want to get is a Pareto frontier out. So when we get to multi-objective optimization, we're not going to find a single solution that says this is the best way to weight all these things together. We're actually going to try to give a spectrum of solutions that traces out a line, and that line that it traces out will be basically will allow the stakeholder to choose among these lines. We can say, You won't be upset as long as you choose along these lines. It's up to you how you want to trade speed for cost. But I can guarantee you that if you choose one of these, you're not going to regret it. Like, regretting something in that case would be like, wow, if I would have just made this other choice, I could have had a faster robot that was cheaper. And I want to just say to you, the stakeholder, that there are no, as long as you choose components along this line, there are no faster robots that will be cheaper. All of the faster robots will be more expensive, or all the cheaper robots. So it guarantees that you're on a trade-off space. And so that's what we'll get to then, how to encode all of that into this big F. For now, we're just worried about one optimization objective, and really, you can just think of the mechanism here. Big F is just a way to make sure little f is positive, because all the selection operators kind of need, well, not all of them, but at least fitness proportionate selection needs this to be positive. And it allows you to also do things like rescale the optimization objective to better balance things like selection pressure. And we'll get into all that discussion again. But for now, just think of big F as a numerical proxy to make little f more convenient. Now, your first question is about the term character, and this is just a common term that is used. It's just meant to be, whereas a trait is a concrete instantiated thing, like brown eyes, a character is the category that brown eyes fit into, like eye color. So it's just, they just sort of fit into a different level of description. What characters do you care about? Well, I care about eye color, I care about shirt color, I don't care about height. So the only characters we put in there are eye color and shirt color. Okay, which eye colors? Brown, red, whatever, and then so those become traits. So the traits are the particular instantiations of the character, and the characters are the things that we care about when we're coming up with what it means to be a quality solution. Does that make sense? So this is more biospeak, which is just inherited by a lot of these evolutionary algorithms. If we were purely in optimization speak, then instead of saying character, I would just say optimization objective. And instead of saying trait, I would say, value of the optimization objective. Yeah.
Are you going to expound on and go back to that idea of generating a line on the trade-off boundary?
Yes, that'll come later when we get to actually multi-objective. So we're not quite there yet, but I do want to just keep you thinking about that, because it will be really important to maintain diversity. Because here, we're actually okay with things eventually stagnating so long as they stagnate at a good solution. But there, we actually want to make sure that we sort of never stagnate, that you always maintain diversity, because we need something that traces out a line. And we'll get to all that when we get to it later. So this is just sort of the baby steps to that. Right, so where to pick up? So yeah, fitness. Any other questions about that? About the mapping, traits, fitness? Yeah, sorry.
What is the difference between the character and phenotypes? I just know the term phenotype.
I see, yeah. So in this case, I would say a phenotype is a suite of traits. So so you say, well, then what is a suite of characters? And so now we're getting into things that aren't quite as well defined. Some people say a characteristic is a suite of characters, and then an instantiation of a characteristic is a phenotype. Some people say a phenotype is both, and you just have to understand by the context. But I would say, for all intents and purposes, a lot of these are semantic things, and a suite of traits is a phenotype. My phenotype is brown eyes and 5'10". And depending on causal mechanisms, you could say that my phenotype, even though it's not genetically determined, for the purposes of the optimization problem, maybe my shirt color is also part of my phenotype, at least in this artificial case here. All right, so any other questions about language? Yeah.
So I was just trying to understand the suite of traits part, kind of escaped me. So what exactly is it with fitness function? Fitness function?
The fitness function maps the phenotype. So I'd say the fitness function F maps the phenotype to some measure of quality. So it takes a phenotype which might have multiple traits in it. So this might be a vector value quantity here. And this functional then turns that into a scalar, which allows us to rank. So fitness basically just provides us a ranking mechanism for dealing with things that we view in a potentially multi-objective way.
So what's my doubt isn't trait just a singular thing associated with a particular individual? So from what I gather, genotype is an instantiation of a chromosome, and a chromosome is associated with just one individual, isn't it?
Well, a genotype is an instantiation of a chromosome. A phenotype are the traits that come out of that genotype. And so there might actually be multiple ways to have the same phenotype. There might be multiple ways to have brown hair. So, or you could refactor code. You could write an algorithm in 2 different ways, and it might produce the same output. The output is the phenotype, but the genotype is the actual code. And then you can have, then a trait is just sort of aspects of, different ways you can evaluate. So just think of a traits as different ways you can evaluate a solution. I can evaluate me in terms of my height, in terms of my education level, in terms of my eye color, and each one of these things becomes a trait. The biological thing sort of has to be linked to something that could be inherited, unless that's where the genotype sort of stuff comes into play. But when we get into these artificial selection scenarios, it's just anything that I can code in a decision variable is part of that genotype, and anything that is a result of that coding that I can evaluate in quality space is what I call a trait.
So it's kind of like you're trying to achieve an optimization of an objective function, and you've got multiple sub-functions within the algorithm, and each one is considered as a trait.
That's right. I mean, you can think of it, there's, I have, the decision variables are my degrees of freedom in my design, but they affect lots of different things. And so I actually don't care about anything in particular in my decision variable. I care about all the things that they affect. And so I penalize the things that they affect, and those are the traits. But then, so, yeah. All right. Yeah.
So in that example that you've given, where you've given that genotype over there, each individual element in that is a gene, right? So if each individual element is a gene, what is the relation of a gene to a trait?
Well, that's complicated because that involves this genotype-phenotype mapping. There may be a simple one gene, one trait mapping, but there doesn't have to be. So you can have, I might have I might have a decision vector that is just, my decision vector might be, sorry, let's see if it's connected this direction. All right. So my decision vector might be a very low dimensional, might only have 2 variables in it. But those 2 variables and how they mix might give me a whole suite of a variety of traits. And so I can penalize each one of those traits differently. And so there might be a gazillion number of decision variables, but in the end, there's only like one trait. So no matter how you mix this thing up, like think about if a trait is like how tasty a soup is. Well, you could put a lot of different things into a soup, but for some reason, people only evaluate it by how salty it tastes. And so there's not like a one-to-one mapping where I can say that I know that if I put these components in, I'm gonna get this saltiness. It's like, well, I could get this saltiness with these components, with these components, with these other components. So there's not an easy mapping from one gene to one trait. The trait is the total collective effect of all of these genes together. There are very few traits that you have, and that like whether your earwax is sticky or waxy, matte finish or sticky, I guess you could think, is one of these single gene traits. Otherwise, things like eye color, height, all these things involve multiple genes, and they involve multiple genes who have subset of genes that are involved in other traits. So there's no easy one-to-one mapping from one space to the other. All right. Yeah.
So the trait and phenotypes are the same thing?
I would say the trait is a part of a phenotype. A phenotype is like a bunch of traits in a, so right now, they're the same thing because we are only talking about one optimization objective. But once we go to multiple optimization objectives, each objective is a trait and the collection of them together is the phenotype. All right, well, let's move on. I think that some of the stuff will become clear if we get back on schedule here. So, all right, so this term fitness, I do want to mention that fitness for us as engineers feels like something that's prescriptive. It feels like we evaluate the quality of a solution, and the quality of the solution affects the representation of the individuals in the artificial evolution. And to some extent, that's correct. To a biologist, fitness is a measure. So to a biologist, when you unroll a population, you then measure how certain individuals' representation changes over time, and then you evaluate the fitness after seeing how things unroll. So for biologists, fitness comes up after. It's a measure of success. For engineers, we often think of it as prescribing success. Like we calculate the fitness, and then that is to drive that. And for most of our optimization algorithms and these design optimization, we can take that perspective. But for those of you who work, you might go on into things that are closer to like artificial life in that literature, then you find, or if you're doing a lot of simulation optimization, then you might find that that you end up taking more of the opposite case, where you actually have no way to evaluate quality until maybe you run your simulations. And then after your simulations kind of become the fitness metric. Or if you're actually running an artificial life experiment where you've got things competing in some virtual space, then you sort of see who ended up getting enough resources if they're directly competing with each other, and then just measure fitness. And so I'm presenting fitness as if we're able to calculate it and then use that fitness to figure out how many individuals go into the artificial evolution. But there are plenty of scenarios where fitness is kind of assessed afterwards as a metric of the process. So I don't want to make it sound like this is the only case, especially because the evolutionary computing literature is so tightly coupled to this like artificial life literature. And we'll talk a little bit about that as we move on. But for now, that's kind of the narrative as we're thinking of this kind of prescriptive terms just for simplicity. And so I introduced a diagram last time, and I'll make it more, I'll make it a little broader and richer here at the end, but it was this kind of simple inverted U where I had selection pressure on this side. And that selection pressure could also be thought of as diversity in fitness. And I want to underline fitness, not necessarily genotype. And then I've got like the effectiveness of the algorithm, of the optimization algorithm. And there was this kind of like inverted thing here where if you have, where I'll put these little arrows, where basically I'm saying here that if you have very, very large diversity in fitness, then you are going to sort of be in exploitation mode where one individual is going to dominate all the other individuals, and very quickly you'll lose that diversity and you'll stop exploring and you'll just kind of be exploiting that area around the one individual that was so dominant. But then if you have no diversity in fitness, then you're not going to have, then all of the movement around your genotype space is going to be effectively stochastic, and you're going to end up going to stagnation just because of the second law of thermodynamics. You just, there's an equal number, an equal representation across the genotypes is unstable, but a representation of only one genotype is stable, and so those stable attractor states are going to end up attracting all the populations until you end up getting stagnation. So it is our goal when we design our selection and mutation operators to try to be in this region here. So we can adjust things like how we interpret fitness or how much mutation we add in, and so on and so forth, so that we try to stay in this region where we maintain some diversity so we can explore the space, but not quite so much diversity that we just take off. Now, anything that reduces selection pressure is in this. Anything, I guess, though, would say that maintains genetic diversity is necessarily going to reduce selection pressure. So it might slow down the algorithm, but that's kind of the cost we pay. So if you go for a very fast convergence, you may just be stuck in a local optimum. If you go for a very slow convergence, maybe you'll be able to find the global, but it might take you a long while to be there. So we need to sort of figure out how to balance those two, and that's what these operators wind up doing. This is one abstract picture, and I will add one here towards the end that makes diversity in fitness and diversity in genotype separate axes so that we can sort of actually visualize how these different operators move around in this kind of diverse space. Yeah.
Just so I have that right, anything that maintains diversity reduces selection pressure.
Right, right. Selection pressure wants to reduce diversity, so if you artificially prop diversity up, you're kind of taking away from selection pressure. You're effectively penalizing. You've got one individual that might be really, really strong, and if selection were to have its way, it would take over the population. But if you're saying, no, I'm maintaining diversity, then what you're going to do is you're taking that pressure, that force away from that dominant individual, making them less dominant. And in general, the message that I kind of want to, that I'm trying to build here is that evolutionary history will greatly constrain how evolutionary processes can optimize. So when we talk about natural selection, we say natural selection is a force of optimization, but it does not optimize. So there are a diverse number of solutions out there, and a lot of them are at local optima. Sometimes they're, they don't have anything to do with, being optimal. I mean, when you turn 30, you start getting root canals. Like, why are your teeth so bad? Well, there's evolutionary history that constrains the optimality. Now, that doesn't mean that there's not pressure towards being better, but the way in which those pressures get washed out by other pressures in the system ends up sort of determining what solutions shake out of these things. And that's why you have to be careful when you use these evolutionary processes artificially, and you also have to be careful of how you interpret things that come out of natural cases. That just because you have 2 arms and 2 legs, that doesn't mean you're the optimal shape for, let's say like a robot that is to do the things that humans can do. It would be great if I maybe had another set of arms, but I'm not going to get them. I'm done. I'm stuck in a region of genotypic space where it will be pretty much impossible for me to ever evolve 2 other arms. But I will exploit, or my species will exploit the space around this bipedal shape, and we'll do as best as we can given the body that we were given from our evolutionary legacy. And it's the same thing with these processes. Question?
So if there is no diversity in fitness, will there be a selection pressure?
No, well, if there's no diversity in fitness, there is no selection pressure, which means that, but that does mean that there still could be changes in genotypic diversity. So you can have no selection pressure, no diversity in fitness, but still have diversity in genotypes, just that those genotypes don't code for anything exciting. But due to random sampling, because there's just limited populations, then gradually your genotypic diversity will bleed out as well. And so again, I'll have a different graph that sort of better captures that. So ultimately, without mutation, everything will lead towards stagnation. So if you have, so ultimately you might start diverse in both, and then you'll rush towards whoever's the at least locally the best choice, and then you'll exploit there. But then once the fitness drops sufficiently low, then gradually this drift will take over, and then you'll get all the same genotype. And if you have mutation, that will add a little bit of novelty in it. But if it's only a little bit of mutation, it's going to limit your novelty, and gradually everybody will be roughly the same. And you can definitely see that when you run these algorithms That's why, like, if you run them with very small population sizes, you pretty much have no selection pressure. Because very quickly, all that diversity washes out. Yeah?
Yeah, I know this is being discussed a lot, but, so in the ideal situation, right, you could have an individual who has a very high fitness, and the diversity could be just the genotypes of two fitnesses, that's it. But this particular individual can dominate and lead to a stagnation, isn't it?
That's right. One particular individual could end up dominating and then very quickly lead you right into stagnation.
So it's possible and ideal. So we need to have some kind of a termination condition, just these many epochs and you stop.
That's one way to do it, but I mean, there's other pressures that we're going to add in once we have this, like, mutation operator, for example. If you have a large enough population and you start with a diverse enough initial condition, then that sometimes can be enough. You can also add other things that prevent things that actually make sure that you maintain niches. So you can have sort of speciation that can happen, so that if you find one local optima over here and another local optima over here, that you can actually keep those separated from each other. So there's a bunch of things you can do to maintain diversity. But they have the downside that if one of your solutions is really, really good, then it's never going to attract these other ones. And so now you're wasting time exploiting an area that's not as good as this other area. But you're doing that in the hopes that the extra searching you're doing might find an even better area somewhere else. Okay. All right, so it's like all of our basic abstract terminology. And so then last time we started, implementing or showing how to implement these things, drew this little diagram where we said, in our GA, we start, we draw an initial population, and one simple way to do that is just to draw a random matrix. I'll do x11 down to xn1 over to x1n down to xmn. And the thought here is I've got m individuals and n genes. And you could just draw that thing randomly, and that's your initial population that's hopefully diverse. Or you might have other reasons to start. You can imagine kind of a Bayesian approach here, where you think that your solutions are in certain regions of the space, and so that's where you start your populations out. For now, I just uniformly scatter everyone out around my space here. And then after your initial population, you go through a selection of R, I'll call them parents. And we discussed there are a bunch of different selection criteria or types here. I gave you the three basic ones. There are additional ones, and each one of these can be specialized even further. There's Boltzmann selection operators and all sorts of other things you can add, but the basic structure of most of them is that, remember that we have this fitness function, big F, that maps the trait, which maps the genotype to a quality, and that quality is, higher is better. So that somehow encodes your optimization problem. And then from there, you can do fitness proportionate selection, which I may have just called proportionate selection last time, but there was some confusion over the difference between this and ranking, and so I'm just kind of making it clear this is fitness proportionate, not ranking proportionate selection, where basically you're going to take your fitness function and you're going to calculate a probability that individuals with this particular fitness get into the parent group. And you just basically can normalize by the total fitness. So it's like whatever that strategy is, whatever that fitness is for that strategy, divided by the total sum of all the fitnesses across the strategies that are in your population. And then that becomes just a probability, and you can use stochastic sampling approaches to then draw your R parents from that. Then the other version is ranking selection. And there are a bunch of different ways to do that, but one simple option that kind of relates to the previous one is you can think of it as a rank proportional selection, where you can think of it doing the same thing I just said with fitness, but you drop the actual fitness value here and replace them with their rankings. And so if you're a good quality solution, you give them a high ranking. If you're a low quality solution, you give them a low ranking. The advantage of that is if there are large differences in fitness, but not very large differences in rank, it kind of squishes those differences in fitness, kind of like the way a log transformation would do if you're doing stats. Question?
Ranking is based on trait or on the fitness score?
It's on the fitness. So you can have multiple traits in here, but we rank by how they map to
So
fitness.
it's ultimately fitness score itself rank, but it's only that fitness scores can vary a lot on the scales. Ranking is not going to vary that much.
That's right.
Okay.
That's sort of the big difference between fitness and ranking. Yeah.
But ultimately, it's just plotting on a line, right? Like proportion, we had imagined it as if your fitness score is more, you're gonna get more of the line.
Uh-huh.
Same thing is gonna, it's gonna end up in the similar way even with ranking. If your ranking is higher, you're gonna end up getting more proportion of the line.
That's right. You'll get more proportion,
How
but
is it different ultimately?
But, well, so exactly the number. So let's say we're worried about that premature convergence. Well, if you had a fitness that was like 1 million, and then the next individual had a fitness of 10, and then 8, whatever, then in that case, you pretty much are guaranteeing the one with 1 million is going to have all of them. But if I do a ranking selection, there's still at least a shot that everybody else will have some bite of the R apple. Okay. And then the third one we talked about is tournament selection. And that involved another hyperheuristic, or sorry, not hyperparameter, that you basically draw Y individuals from the population, and you pick the best one out of those Y. So you draw Y individuals randomly, and you pick the best from that random group. And then you do that again R times. And so that is another way that sort of inherits some of the features of ranking selection. The dynamics of these change, but basically these two are meant to be just alternative ways of reducing the effect of large fitness differences in this one. The original GA was kind of like based on fitness proportion selection, and then some clever computer scientists said, you know what, let's just make it all based on ranking. There's two different ways we could do that. We either rank them and make it ranking proportionate, or we do a tournament. And there are slight different dynamics in these, but really these two were like kind of in a family together that's sort of separate from this one. Yeah.
Yeah, my question is the tournament selection. Just like we take Y individuals which are all good at fitness, and then we select one from them which is the tops them, and then we take another Y which is like they're all bad at fitness, but even though they're all bad at fitness, comparatively one among them must be
That's right.
higher. So in this case, are we getting like bad individuals into the selection?
Very often it is very possible that you might draw but that helps guarantee that. So that sort of tournament selection is meant to be even, like if you were thinking about this, fitness proportion selection strongly favors really high fitness. Ranking selection is kind of intermediate. Tournament selection, everybody's got a shot. It's sort of like, if you think about sports, any sport that has, a tournament at the end of it, you kind of give up. The rest of the season doesn't matter as long as you get into the playoffs. That's what this tournament selection is. It's like how well you actually rank doesn't really matter so long as you get into the tournament, because you're right, you could play some other guy that's just as bad as you, and that might actually allow you to survive for a pretty long time. So tournament selection is sort of amplifies the representation of the low ranked solutions. Ranking selection is kind of gives everybody sort of an equal shot, and then fitness proportionate gives an extra amplification to the ones that are high fitness. And then, of course, there are scaling methods that, especially with these two, you can adjust. You can add kind of a sigmoid or whatever you like. You can play with this fitness function so that you can further massage these differences. But that's the basic structure. So that's where we were last time. We got all those. And now the next step, we've got our parents, and so we need to do the reproduction step. And reproduction is going to have these two components, crossover and mutation. And you can think of crossover as breeding or blending, interpolation and extrapolation, and you can think of mutation as adding variation. And this, so far here, the encoding that we chose really didn't matter that much. Whereas in here, this is where encoding starts to matter. Because how you're encoding, I mean, that's what we would talk about, like in animals, or actually just across all the domains of life, we have chromosomes, and those chromosomes are what determine what crosses over. And where those chromosomes pinch over is what's kind of says what genes are always going to stay together and what genes have a chance of separating. So the way you encode your decision variable starts mattering when you start crossing things over. And the way we implement mutation is we often mutate at particular sites. Certain genes get mutated. So whatever you spread out into a gene will then often can be a direct target for mutation as opposed to like an indirect target for mutation. So we'll see that as we implement these. And then we're about to go into how to write those, but then after we do that reproduction, we check to see if we've reached termination criteria. If yes, we end and report a population. So I'll write population there. And if no, we go back and we reselect the parents. And the basic idea here is our old had M individuals. Our new has R parents, which you can also think of individuals. You can either think of this as overlapping generations, or you can think of R asexual breeders. So they were just directly copied. But then those that managed to survive to here also gain the ability to then make offspring through crossover. And there's M minus R offspring. So these are the offspring, and these are the parents. And so the old population has M, the new population has M, with R from the old plus a new set of R that have been crossed over and mutated. And that's the way the algorithm works. Yeah.
So you can reuse the parents, right, multiple times? Same parent multiple times?
What you mean in these?
In reproduction.
Yeah, right. Well, I mean, in most of these selection criteria where you're drawing new parents, it's with replacement. So you might draw from the, you don't, once you've used them, you don't put them aside. And so it's possible that these are parents, could be a direct copy, but you might have actually, one parent here might now have multiple representation here. So there can be changes in distribution. I don't want to say it's a direct copying necessarily, but that's why I think it's more like asexual reproduction here. So you could have one individual that, due to their high fitness, duplicates itself in 3. And then that effectively gives it a greater chance of creating offspring that are similar to it, because now those 3 individuals get a chance to mate, and it's more likely that they'll mate with themselves, or they'll at least be a half of the matings that go on.
But now I assume I have R unique parents. When they reproduce, RI could be used 5 times, right?
That's right.
First mate is every time its mate may be different, but it could mate 5 times.
That's right. It could mate 5 times. Yep. Now you could definitely, this is the general overview of a genetic algorithm. You could certainly make, these are kind of pluggable. So you could put in your own selection, mutation, and crossover that prevent that if you felt that was important to, say, maintain diversity in your population. But the kind of prototype is, yes, you could have multiple. Yeah.
So every time you go back to your initial population, that's like trying to do another generation, getting another set of the
Yeah, well,
production.
when you go back up here, the new becomes the old.
Oh, and then you
Yeah,
cycle through.
That's right.
And then for the terminating criteria, we haven't talked about that yet.
Oh, well, that's, so I mentioned it briefly. So the termination criteria are kind of up to you. They're typically, say, number of generations is be one criteria. Number of function evolution or function evaluations might be another criteria. It might be very costly to evaluate the optimization objective. Or some convergence criteria. So it might be that once you don't have more than a certain tolerance of fitness difference across into the population, you stop. And that's sort of up to you.
Gotcha.
Okay. All right, so let's see these operators. So let's start with the crossover operator, which is the kind of numerical version of sex in these populations. And so there is a new hyperparameter that we do, we call PC for probability of crossover. And so basically what we're going to do is we're going to take random pairs. Maybe I'll write that. So we'll choose a pair of our parents, and we just draw them randomly, uniformly. And so there are some questions in the muddiest points about what I mean by uniformly. All I mean is that every parent has an equal probability of being drawn. So the distribution across parents in probability mass is uniform. So every parent in the R parents could be chosen. So we draw one, and then we draw another. So now we have a pair of parents. And then for that pair of parents, we flip a coin, and with that's weighted with probability PC. And so they cross over with probability PC. And I'll just say PC is between 0 and 1. So again, stochastic sampling, I just draw a random number between 0 and 1. I compare it to PC. If it's under PC, then they cross over. If they don't come up for crossover, I put them aside, and I redo this process. So the only ones that that actually get to cross over are the ones that are in this potentially small group. And I just do that over and over again until I get enough of these crossovers. So then there are different types of crossover that I can implement. The simplest type is 1-point crossover. And in that case, if I have a genotype, then I'm going to depict, these are two genotypes next to each other. And for now, I'll draw, imagine that this here are N genes, and these are two individuals. So these are our two parents, and they have N genes. And another hyperparameter is where I'm having my crossover point. So I just say ahead of time that I'm going to cross over between genes 4 and 5 or something like that. So I set a crossover point. Let's say it's right here. And then I'm going to get two offspring out. But those two offspring are going to be a mix of the head of one and the tail of another. So the offspring that I get out will also have N genes. So there's my N genes. And they also have the same crossover point. But if I were to say, I'll crosshatch these ones this way, and maybe I'll put circles or squiggles in this bottom one here. And then so these ones here their encodings get switched so that my two parents stay in their form in the parent population, but the two offspring that come out are now mixed so that they have this part of the encoding from parent 1 and that part of the encoding from parent 2 and vice versa. Yeah.
So what do you do if that child is not feasible? You just throw it away? Thank you.
Yeah, right. So, excellent question. So, generically, what would happen is you'd have this if it was unfeasible, then well, okay. So there's two different types of non-feasible. They have really, really low fitness, in which case just roll it into the next population, and then just let selections select it out. But that's pretty slow because if this. And so the other thing you could do is if it kind of depends on how true you want to be to this probability of crossover. You could just say, sorry, you guys might have made it, but certain things in the certain before implantation things weren't viable, and so you can think of this as like a failed crossover, as if, and so in which case if this doesn't meet your it doesn't fit in that set X, that subset of Rn, then you just you handle it as if they weren't selected for crossover and redo this, which is also slow. Now, what people like to do is say, well, actually, can we, if maybe in our mutation and our crossover and our selection, if we have a way to maintain diversity, then maybe we make sure, say in the crossover, that we only select parents that are close enough together so that when this happens, we're almost guaranteed to get a feasible solution out. So you can sort of create, and this is like where the speciation comes into play. So if you can, if it happens to be, you can penalize. So you can imagine making this probability of crossover like a function of their Euclidean distance in decision space. And so if they're very, very far away from each other, we say it's very unlikely that those two will mate. But if they're very close to each other, they will mate, and then that helps guarantee that you get feasibility out of this. You can also hopefully create your encoding and your crossover point so that maybe you don't cross over in the middle of something that could really screw something up.
So it seems like you'd have to, at times, get super creative with this crossover function.
Yeah.
Because for example, if your genotype has to be a valid permutation, for example, this 1 or even 2 point crossover is never going to produce, or almost never going to produce a feasible solution.
Sure, right. And actually that's the reason why as we'll talk about, we get the multi-objective. The genetic algorithm with the crossover operator, when the GA came up, there were I think 2 or 3 different other genetic algorithms that were inspired by evolution but did not have a crossover operator. And so you can get rid of crossover like we do in the second part of the homework problem, and that is a totally feasible thing to do. You can say, this is just never something I want to do because I can't imagine this will ever work out well. Now there are other encodings. When we get to genetic programming, then you can imagine well, if I just encoded each one of these as a line of code, this would be a terrible idea because I might have a for loop here and a while loop here or something like that, but I have a couple of print statements and a for loop here, and now I'm going to cross, it's just not going to work. But there are ways of encoding syntax trees to guarantee that when you cross over, it'll always be, at least it'll be executable code. And there are also different programming languages you can use that guarantee that you don't kind of have any baggage that lives beyond a single directive. So, you do have to be very creative, and sometimes you just don't worry about crossover because there's lots of other good stuff going on for you. This really makes sense for numerical problems where you want to do this interpolation, extrapolation thing, like the kind of the problem I have you do in the homework. But there are plenty of other problems when you get multiple genes where, this can be not a great thing. All right. Yeah.
I think you also mentioned in the first class is another possible solution, forcing the children, projecting them back into the correct space, or is that a different issue altogether?
No, yeah, so that's something you can come up with other ways to maintain feasibility. And so if you would if this is out of your set X, your subset of Rn, and you want to run another nonlinear optimum like an NLP or something, if you want to run a nonlinear program that projects them back into your space and you can pay the computational cost, that's fine to do too. Yeah. Uh-huh.
In real life, is EC close to 1.5?
In real life, you know, I don't really have like a good heuristic for what the crossover probability will be. I mean, it's sort of going to be application specific. That's why there are these hyper heuristics, which you sit on top of your genetic algorithm that could choose different values for this.
When a man and woman marries, right?
Oh, I mean, I see real life here. Well, think of this as not, think of this as wrapping up into it. This is like two people meet and they decide to take things to this level, and they're both fertile, and they have offspring that, you know, survive to reproduction. So you can add all of those ands together, and this PC might get very low. Yeah.
So you did a tailwise swap there, right?
Uh-huh. Well, I mean, I don't know if it's headwise or tailwise, but I mean, I just swapped. I left these in, it's kind of arbitrary which one I say I left, because I can just sort the individuals differently.
Gotcha. So it doesn't matter which one you leave and which one you swap.
That's right.
Okay.
That's up to your implementation. But that does get to the other, so there's also 2-point crossover, which is another way to do with things, or multi-point crossover. And so you can imagine I could have lined them up as before, but I might have drawn 2 crossover points, and I swap the head and the tail, but I leave the middle part alone. So that's another way to do it. And then there's uniform crossover, where in that case, maybe I draw crossover, I'll do CO positions uniformly, and then do the crossover. So in this case, you might just say, I want to do a 3-point uniform crossover. So whenever I get a crossover, I choose 3 points at random, and then I cross, leave alone, and so on down the line. So there's lots of different ways you could do it. Maybe you make the choice informed by the particular problem you're working on. If you have, basically the idea is if you have a really, really long genotype, you probably want to do more than a single point crossover, because you're just not going to get that much mixing. Yeah.
Wait, so do you say you draw the crossover points at random, or?
For this type of crossover. For these, you might set them ahead of time.
Okay.
All right, so those are the basics behind the simplest kind of crossover operator. Are there other questions about that? Lay out your genotypes, swap, pick a point, and then leave things on one side of the point alone and swap the stuff on the other side of the point. Yeah.
Yeah, so with regards to how we choose the parents, so this is chosen, when you mean uniform, it's totally random, right?
Yes.
Not based on some sort of a distribution or something in
Right, I mean, I guess you could think of it as a uniform distribution across parents. So every parent has a 1/r probability of being chosen.
Okay, and once a parent is chosen, is there a replacement? I mean,
Yes, usually the way these are implemented is that after the crossover, they both go into the mating pool and they're given another opportunity to breed. Yeah. Yeah.
So a parent can survive multiple rounds of production.
That's right. In this sort of generic version here, again, your mileage may vary and you are free to make these changes, that these things are all pluggable, so to speak. Okay, so the other operator, mutation. And so that introduces another hyperparameter, which I'll call PM. And the simplest way to handle mutation is you've got all these offspring that you just generated. So you could do this right after the crossover or after you're done with your crossover. So if it wasn't clear, that crossover is going to generate 2 offspring every time. And you just keep doing that until you fill out the N-R offspring you need. So then of those, the simplest way to handle mutation is you can just iterate over all genes, so all positions in the offspring. And then you just, with probability, and so then for each gene, with probability PM, you mutate. And so what do we mean by mutate? So I'll put mutate in quotes. And there's a bunch of different options you can do here. The simplest thing, if you've got especially a discrete genotype, is a uniform mutation might be that you've got some gene XJI, so let's say this is the jth individual, this offspring in the ith gene, and regardless of what used to be there, I'm just going to take, draw uniformly, and so this case, this is continuous, but you can think of continuous or discrete, whatever makes sense for you. But I'm just saying, all right, at this gene, I don't care what it used to be. I flipped a coin, it was targeted for mutation, so I'm going to replace it with a random value between here and here. And then that will be that offspring. Now you might say that's a little harsh. That's taking that, that you're taking something that might have been okay and you're completely moving it around. And so another option is, non-uniform. And you can do lots of clever things here. So one, relatively simple idea would be to, well, let's take the next XJI is going to still, I'm going to use the uniform distribution, but I'm going to center it around the previous gene and sort of set lower and upper bounds around that one, so it's kind of like a little confidence interval, and I'll shrink that interval over time. So I could say it's my previous XJI minus, and then I'll create some step size, or whatever you want to call it, I'll call it D naught, and then multiply it by big T minus T over T, and then I've got the flip of that on the other side. And so this will be plus D0, T minus T over T. And then I'm going to define T as my termination time step. And I'll define T as my current time step. And then D0 is just some parameter that is greater than 0. So the idea here is early, I'm centered on whatever the previous value is, and I then, since it's early, then this will be a large number, so this will be close to 1. So I now will then draw a random number centered on the previous number that is, but drawn from the largest range. But in the next generation, so that's on time step, so maybe instead of time step, I could have written generation for that. So in the next generation, then that interval is going to shrink a little bit, and then a little bit more, and then a little bit more, until when I'm at the last generation, I basically have no mutation left over. And so that is one way to kind of say that we're going to try some kind of crazy exploration initially, and then over time, we're just going to exploit. We don't want to screw up a good thing. We're hoping that the genetic algorithm is going to get us close to a good solution, so we want to be able to jitter a little bit to try some new things out, but we don't want to go crazy. And so that's one way to do non-uniform mutation. But there's a bunch of other ways you could imagine to do mutation, but they're all in the spirit of this. You either knock out the whole thing with a shotgun, or you start, you might start in a little more conservative way and then get more conservative over time, and then come up with your own. Yeah.
What if your PM is high and you mutate a lot? What is the risk of that?
Well, I mean, this PM is sort of up to you. So, and I would say normally you probably want to set this low, because you've got a lot of genes, so there's a lot of opportunity for this to happen.
Like define low, like.
Well,
25%, 25%, what's low?
Oh, I mean, I would say, less than 1%, so like pretty low, because, hopefully the rest of the evolutionary algorithm is going to be doing a lot of the searching for you, and you don't, if you set this too high, this just becomes a stochastic search. All right. Yeah.
Is there a chance we could change this PM or PC while we are iterating over? Like initially we keep it high and gradually.
Absolutely, yes. So I meant to mention that a little bit earlier, but yes, you can adaptively change these things. In fact, you can estimate your current selection pressure. And if your selection pressure seems to be getting a little high or a little low, you can crank these things up and down. Yeah?
Are there any like hybrid approaches that combine genetic or evolutionary algorithms and like simulated annealing? Do you have like, are there like cooling schedules for mutation or stuff like that?
Yeah, I mean, I would say that, so we will talk about simulated annealing, but I mean, I would say that there certainly are ways that you can set schedules up like this that look like simulated annealing, that have the same spirit of simulated annealing. I would think it would be overkill to combine this with simulated annealing because they do a lot of the same things. But what's more common is to combine this with something even simpler like a gradient-based approach or something like that. So you use this to get in a course area, and then you kick off a gradient-based approach that sort of define the bottom of the barrel or something like that. So those are the kind of the hybrid approaches that are more common. But my guess is if you try to combine with simulated annealing, the simplest thing would just to leave out the simulated annealing, but put your cooling schedule in here. But we'll get to that. And so simulated annealing is like a non-population-based, nature-inspired approach that happens to do really, really well. Kind of similar to tab research. All right, so just to kind of like to wrap things up here, so I wanted to, there were like 4 hyperparameters that we've sort of talked about, and we've hinted at a couple of more. So we have these parameters which define the performance of our algorithm, the population size, the number of genes as well, the number of parents, the crossover probability, the mutation probability. Now you might think, well, how do I set those? And of course, I mentioned you could wrap another optimization algorithm over that, but it does help to have kind of a conceptual model. And so that model, I put a sort of a PowerPointed version of this online. But if we draw a different diagram, that might be, at least for me, it's helpful when I try to conceptualize what these changes in these parameters do, and then how I can do things like these adaptive schedules. And so if I put on this plane here, if I forget about the efficiency of the optimization algorithm, and instead I put diversity in fitness here, which is equivalent to my selection pressure. And then if I put genotypic diversity here, so sorry for the handwriting, but that's supposed to be genotypic diversity up there, then this sort of turns into a space with 4 quadrants. So down here, this is stagnation or fixation. And so you can think of this like I have no genotypic diversity, everybody is identical, and because everybody is identical, I have no fitness diversity. Evolution cannot move. There's just nothing that you can do there. Now, I can mutate, and so you can imagine that there is some mutation force that is sort of positioned mainly upwards towards, so I'm going to call this mutation force. And it's primarily when you mutate, it's not doing it in any fitness specific way, although I suppose you could try to mutate in directions that are more, based on some model of fitness that you've built up over time. But in general, the basic idea is mutation primarily mutates in genotypic space, but as you mutate your genotype, then you're probably changing your fitness as well, creating more fitness diversity. So it's tilted a little bit here. Now, as you've got selection pressure, that is moving more, say, in this direction. So I'll call this selection pressure. So the strength of that pressure is kind of along this coordinate, but the direction it moves things is down this way. So with more selection pressure, you're going to get a greater tendency to reduce your diversity and fitness and reduce your genotypic diversity. And meanwhile, while all that's going on, then you've got constantly drift, which is pulling you toward this stagnation point. And so, as you can kind of imagine, drift, these vectors are getting closer and closer together here, then drift is strong, so here, so this is strong drift, and this is weak drift out here. And another way to view that is you can think of, this is the region where drift dominates, and this is the region where selection dominates. And moreover, you could even go farther and say that up here, where you've got a lot of genotypic diversity, then you're in an exploration mode. It's kind of like in this direction, and down here, you're in exploitation mode. This direction. So you've got these regions of drift, exploration, and exploitation, which are dominating this space. And so if you increase your population size, what you're doing is you're basically taking this cross point of these two red lines and pulling it down here. Increasing your population size increases the amount of space where selection is going to dominate because you're going to get more diversity. Decreasing your population size is basically going to mean drift is going to dominate the whole plane. And so your initial condition, when you set an initial condition, that initial genotypic diversity is going to put you hopefully up here. But then as selection acts on you, if you've designed things well, you'll move in this direction. So you'll explore for a while, you'll exploit, and then gradually over the exploit, you just can't help it, you'll eventually drift and you'll stagnate. And so that's kind of the optimal way to do things. So when you're trying to think about like, what is my mutation operator doing and all that sort of thing, well, mutation is constantly moving you up, but mutation is going to also screw up any fitness gains you've got as well. So you can't just say I'm going to answer drift by adding mutation. So in the end, there's really a speed accuracy trade-off here. If I increase my number of individuals, it's going to take me a very long time for the algorithm to run, but I will not need much mutation because drift will not be much of an issue. But if I decrease my population size, the algorithm will go very fast. But if I want to make sure it's not just all about drift, I'll need to increase my mutation. And by increasing my mutation, I basically prevent myself from ever finding high resolution solutions. So it's the speed accuracy trade-off where if you want high precision solutions, you have to have a lot of individuals and a low mutation rate. If you want a fast algorithm, you need few individuals and a high mutation rate. And that's all because you're always fighting this drift. So there's a cleaner version of this that I drew online, you can find there, and I'll tack it on to the end of this when I post these. But that's all the time I've got you for. So if you have any other questions, you can come up after, and we'll start talking about moving on from the GA next time around. So now you should have enough info to give a shot at that, making those two simple algorithms in part A and part B of that second homework.